So our next speaker is Tony Wyss-Coray of Stanford University, and he'll be speaking on immunomodulation to promote cerebrovascular and cognitive health. Tony.
Can you hear me? Is this working? Yeah? Turned on? Okay, I want to really thank also the Kathy and the Allen Frontiers Group, as well as the American Heart Association, for allowing us to do this exciting project that, like Rusty said, also in our case brings together people with very different expertise. I like to start with this slide here and see a show of hands for people who ever had a problem finding a car in a busy parking lot like that. Okay, a few people. As you get older, it becomes harder to create what we call these spatial maps, not just create them, but also retrieve them. Whereas if you're young, you don't really know what I'm talking about here. You can have your radio on, talk to your friends, go shopping for a few hours, and you come out and you never even think where you parked your car. You just automatically walk there. But we know as we get older, this capacity becomes impaired. It's actually the case also in animals, in dogs, in mice, and in monkeys, as Rusty showed in his project. So what happens to this young brain that works so beautifully well and can store information and retrieve it and becomes susceptible to degeneration at old age? We know we lose synapses, we lose cells, mass shrinks, and really, the brain does not function as well. A few scary numbers. At age 85, roughly 32% of people have Alzheimer's disease. And if you add other dementias, this number may be even a little bit higher. 81% of patients with Alzheimer's disease are above 75 years of age. And it's estimated that just in 10 years or so, there will be 75 million people with this disease. Now, we know that in a very few individuals, autosomal dominant mutations can cause this disease. And you see this in the upper left here. Mutations in these genes cause the disease. We also know that there's many GWAS hits, so mutations or rather polymorphisms in the genome that increase your risk to get the disease, very slightly so. But they're very common. But really what the key factors are, the key risk factors for getting Alzheimer's disease is age, in addition to vascular risk factors. So these are really the key factors, the elephant in the rooms, that make our brains more susceptible to degenerative diseases, such as Alzheimer's disease. And just a brief vignette on this. This is a post-mortem study from Dave Bennett and Julie Schneider, who Dave Bennett is actually part of our consortium, that analyzed 1,000 brains roughly from people who passed away at old age, 84 to 93, and they looked at these brains and they observed in up to 70% of individuals pathological changes in the vasculature. And this number is even higher in people with Alzheimer's disease, where it rises up to 80%. So it tells us that there is massive pathology in people of old age, just dying either from disease or without cognitive deficits. So this formed really the basis of our overall hypothesis. We want to understand how this young brain, influenced by age, by inflammation and vascular injury, becomes susceptible to cognitive impairment and dementia. And we assembled a team. Marion Buckwalter, the co-investigator, is actually here as well. Chenghua Gu from Harvard, she's also here. And then we have many other people at Stanford, as well as I mentioned David Bennett at Rush, who came together to tackle this question. And I show you now 3 vignettes of the types of data behind the thinking of this hypothesis, and then at the end, the specific aims that we came up with. So the circulatory factors and vascular injury modulate cognitive function. I'm showing you first a plasma proteome changes dramatically with age and modulates cognition. that endothelial immune receptor VCAM1 regulates brain inflammation and cognition, and then that a mild vascular injury can result in delayed cognitive impairment and recruitment of immune cells to the brain. So in this experiment, and this actually started more than 15 years ago in my lab, where we tried to see if we can understand brain aging by looking at factors in the blood, by basically probing the proteome of the blood plasma, of the liquid portion of the blood, the plasma. And this has increased in most recent rendition here to over 4,000 individuals, healthy individuals where we had plasma from 18 to 95 years of age. We were able to measure close to 3,000 proteins with a platform that uses modified oligomers, so oligonucleotides. And you see here such an oligonucleotide, they call these aptamers, bound to interleukin-6. So they function like antibodies, but they're based on DNA, modified DNA elements. And with this, you can specifically recognize thousands of different proteins. And this was a collaboration with Nir Barsala and Sophia Millman and Einstein College, as well as Adam Butterworth of the interval study, and with Somalogic, who actually ran the analysis of these samples. And what we found, if you look at this here globally, is dramatic changes with age in the composition of the plasma proteome. So here we look at 3,000 proteins, many of them involved in signaling activities, but really this is a broad coverage of the overall proteome. And on the x-axis, individual people, so this is cross-sectional, from 20 to almost 100, and then the 3,000 proteins on the y-axis. And what you notice is, again, first of all, these changes are not linear. There's actually waves of aging that we can describe. But importantly, what you find also is that there are factors that are expressed at low levels, shown here in blue, that increase with age, many of them in inflammatory injury pathways. There are other proteins that are at higher levels when you're young and decrease with age, and they tend to be sort of maintenance proteins, that are involved in developmental processes. So with this information, we can actually also build a chronometer or a clock, if you will, such as Stephen Horvath has shown for the methylome. We can look at changes in the proteome, and we can train an algorithm to look for changes with age and then predict the relative age of a person, and this is shown here. So on the x-axis, you have the actual age of a person, and we calculate, based on the top proteins that correlate with aging, the relative age of these individuals. You can see there's a nice prediction, and the deviation from this axis, the so-called delta age, is really what you're interested in, and asking, is this related to biological aging, to functional differences in how we age? And that seems to be the case because we can look at many different functional capacities in these individuals, and we have actually many of these here. So each dot here is a functional measurement in roughly 1,000 people from the Einstein cohort. And we see a strong correlation of the delta age with cognitive function as well as physical grip strength. So if you look biologically younger, if you're above this curve here, if you're below this curve here, you tend to have higher cognition and better grip strength when you're old. And if you're above, if you're a red dot here, then your cognition tends to be lower, your physical strength lower at old age, suggesting that these changes that we see in the plasma have actually a biological correlate. They're not just correlating with time, but they actually correlate with function. And this relates very much to a type of revolutionary experiment that Tom Rando first did called parabiosis, where he studied muscle stem cell aging and asked the question, do these stem cells age because of cell intrinsic changes or because of changes in the environment? And he paired young with old mice and could show that an old muscle could be regenerated. This was a study with Irina Conboy in 2005, that an old mouse exposed to a young mouse, to young circulating factor, can regenerate an injured muscle almost like a young animal. And this led to a collaboration with Tom's lab. Tom is also at Stanford and is my lab neighbor. And we explored this in the brain and could show indeed that the brain also benefits, an aged brain benefits from exposure to young factors. And this is shown here. So either parabiosis or plasma transfer can have beneficial effects on the brain, but also on many other organs. And just to give you a little bit more detail, we were able to actually show that you don't need to suture animals together. You don't need a shared circulation, but you can simply collect the plasma, the soluble factors in blood from a young mouse, repeatedly infuse it into an old mouse, and that leads to an increase in neural stem cell activity, increased synaptic plasticity, increased neurophysiological activity, attenuation of inflammation, and improvement in memory function. At the same time, if you do the opposite, if you take plasma from aged animals or old people and infuse that into a young mouse, you get the opposite. You get reduced stem cell activity, reduced plasticity, an impairment of memory function, and an increase in neuroinflammation. And here actually also you can use human plasma, put it into a mouse or young mouse plasma. So together, this all suggests that there are factors in the blood. The changes that I showed you can actually modulate aging of the organism, and specifically of the brain. And that bears out if we look at individual cell type, and just show you here as an example vascular cells. So these are brain endothelial cells that we isolated just first to look at young versus old cells. So if you isolate endothelial cells from the brain or from any other tissue and you use single-cell transcriptomics, they spontaneously cluster into these three different domains: arterioles, capillaries, and venules, ACVs. And when we look at changes, pathways that change with age, we see that they actually age differently. In arterioles, you see changes in cell adhesion, mechanical stimulus cell cycle arrest, capillaries, blood-brain barrier transport, ATP vessel remodeling, and in venules, DNA repair and RNA splicing. So there seem to be different programs of aging in the vasculature, and there are some common ones as well. But what happens now if we expose a brain from a young mouse to an aged environment? We can indeed change the signature now, and this is shown here. So in the x-axis, we look at what I just showed you, aged versus young. So these are genes that are going up with aging in this direction, genes going down with aging in that direction. And here we look at a young mouse that is injected with aged plasma, genes going up with aged plasma, genes going down with aged plasma. And you see there's a significant overlap of genes that are going in the same direction, suggesting again that there's factors in aged plasma that can mimic aging, that can drive the transcriptome of these endothelial cells towards an aging phenotype. And maybe that starts to help explain why we see these prominent effects that are relatively acute when we change the composition of the blood or the plasma specifically. So let me get to the second point. Endothelial immune receptor VCAM1 regulates brain aging. So if we think about this, how does it work that these factors in the blood exert these effects on the vasculature and beyond on the brain? And there's multiple ways to think about this. So proteins may be actively transported into the brain through transport receptor-mediated mechanisms, such as transferrin, for example. There may be openings in this blood-brain barrier that allow factors to directly have access. And then it's possible that factors directly modulate the activity or the transcriptome of endothelial cells, what I just showed you before. So we focused a bit more on one specific aspect of this possibility. And this is a protein called vascular cell adhesion molecule 1. And it came up out of one of our early proteomic screens where this was one of the top proteins that changed with aging in plasma and was related to the vasculature. So we decided to focus on that, and it has indeed been shown that soluble VCAM1 is a good indicator of vascular injury, not just in the brain, but also in the heart. So it's constitutively expressed in the endothelium throughout the body. It can be shed constitutively to form a soluble component. It increases with age, stroke, and in response to systemic inflammation. And it mediates adhesion to leukocytes. So leukocytes can bind to VCAM1, and that can facilitate the migration of these white blood cells then into tissues. It's not a guarantee, but it is a prerequisite for them to enter tissue. And it's actually targeted in multiple sclerosis with an antibody against the cognate receptor of VCAM1 called VLA4. So the test to look at brain function or higher cognitive function in mice that we use is sort of a modified parking lot test. And we basically train mice on this big round table here to find a single hole where we mount a tube underneath and where the mouse can escape. And so similar to how we use spatial cues such as a storefront or a lamp on that parking lot, the mice use this checkerboard, the triangle, the rectangle to make a spatial map and to remember where is that escape hole where I can disappear from this open, scary environment. All the other holes, they would fall to the ground. And let's now look how so I just have the statistics here, and this is actually a very close collaboration with Marion Backwalter. So we use this to just first look at a young mouse, and you see here over 5 days, 4 trials per day, the mouse is trained to find this hole, and initially we rescue it after 90 seconds. It takes a while, but then over time they learn this task and get really good at it. But let's look how an old mouse performs. They basically have a very hard time figuring this test out and remembering where the escape hole is, and there's a huge difference compared to a young mouse. But if we treat now with this VCAM1 antibody, we can almost completely rescue the performance of these old mice. So treatment of antibody of these old mice for 3 weeks, given systemically, somehow can restore and rescue this impairment in old mice and can make their cognitive function improve to almost young levels. We can actually show that compared to young, so in the aged mouse, you have these reactive immune cells called macroglia, you have changes in astrocytes and neurons, and you have less stem cells. And when we use this antibody, we can recover many of these aspects. We can actually reduce inflammation with the antibody in the brain. We can increase neural stem cell activity. And as I showed you, we can also restore cognitive function. So this is one direction that we want to pursue in our project. And then lastly, I want to show you an example of a mild vascular injury, a mild stroke that can result in delayed cognitive impairment. And so here, this is a model that Marion Backwalter developed. So you give the mouse a mild stroke that leads to inflammation and neurodegeneration, and then the behavioral readout is this Barnes maze again. And when you do this, initially, the mice that are stroked, shown here in red, show an impairment about immediately after the stroke, sort of 2 days after the stroke. You can see that the control mice do well. They find this escape hole quickly, but the stroke mice don't. But then they recover. After about a week or so, they perform almost like they were not stroked. But look at what happens if you look a few weeks later. So this is 6 weeks after stroke. And here what we see now is that the control mice learn the task again, and they do really well on the 3rd day here after testing them again 6 weeks after the stroke. But the stroked mice can no longer learn this task. So there's something that happened in these 6 weeks that even though they seem to be recovered immediately after the stroke, there is a slow degenerative process that occurs that is actually accompanied by infiltration of lymphocytes into the brain, specifically B lymphocytes. And they seem to have a direct effect on impairing cognitive function and contributing to this cognitive deficit, because Marion can ablate these cells and then recover cognitive function again. And so one possibility is that this is related to VCAM1, what I showed you earlier. So based on these the background information on these data, we propose to do the following experiments. We actually assembled a cohort, or we were starting to assemble a cohort of humans who either had a mild stroke or at risk, have strong vascular risk factors. We're planning to recruit up to 400 individuals. We will test them yearly for cognition. We do brain imaging. We do extensive proteomics, and we look with RNA sequencing. Actually, we will go beyond immune profiling using CyTOF to directly do RNA sequencing of cells, both from the CSF as well as from plasma. And then we also have a group of brains in collaboration with Dave Bennett from people who passed away from mild stroke or who had vascular dementia. And we look by single RNA sequencing at the changes in these brains to come up with key proteins and pathways that are altered in people with vascular changes and dementia. And then we want to select these and validate them in models, in mouse models. And specifically what we're going to do is we're going to use the stroke model that I showed you, but we are also going to increase vascular risk factor with diet, and we include age. And we look specifically whether the transcriptional signatures that we find in these animal models are similar to what we see in humans. We will do modeling both with iPSC-derived cerebrovascular models, similar to what Rusty described, as well as brain organoids. And we will also do modeling of the neurovascular unit to try to understand and validate the changes that we observe in humans and go back and forth iteratively. And then the ultimate goal is to pick individual factors and move them towards the clinic and test them. And we believe VCAM1 is a good initial candidate to pursue and study further in these models here, validate that it shows similar changes in human brains, and then potentially move it forward to the clinic. So with that, I'd like to thank you, and I'd be happy to take a question if we have time.
We have time for at least one question, and go ahead.
Tony, very nice. Just a question, two related questions. Is the effect of the VCAM monoclonal antibody on membrane-bound or soluble VCAM1, as far as you can tell? And then secondly, what if you use natalizumab, either a murine equivalent, or if you look at patients that have been treated with natalizumab?
Right. So the first question is VCAM1's effect on the membrane-bound or the soluble form. We can't answer the question completely. We did inject H plasma that lacks the soluble form. That still causes deficits. We also can genetically delete VCAM1 in the vasculature and can rescue the deficit. So we believe it's probably the brain vascular VCAM1, but we don't understand exactly what the mechanisms are. Is it a signaling event? Is it binding of immune cells to the vasculature in the brain, sort of a slowing down? We know they don't go in. So we still have to figure that out. And then the question whether antibodies to VLA4, the binding partner of VCAM1, are efficient. We did indeed see with VLA4 treatment a rescue of the inflammatory phenotype. So I think there is certainly a partial effect with VLA4 as well. But maybe with the VCAM1 you actually target the endothelium directly, and that may have a beneficial effect.
We have time for one brief question.
So you recently showed, I believe, that TIM2 is one of the main factors that promotes the anti-senescent phenotype. Do you know if this acts, for example, by inhibiting MMPs, which are part of the senescence phenotype? And conversely, in that aged plasma experiment, were MMPs outliers that were associated with contributing to the aging effect?
Yeah, so I think it's more complicated. So we described an inhibitor of metalloprotease called TIM2 as having a beneficial effect, as being part of sort of this cocktail of good factors, but there are many others. I was very excited by the talk earlier about the extracellular matrix and the composition and your presentation. I think there is definitely changes in the matrix, and when we look sort of at these global changes with age, we see very strong pathways that point to extracellular matrix. But which components, I would be very interested in talking more.
Thank you so much, Tony.
Thanks.